All right, good morning, everyone. I'm Gary, the Director of Marketing for Abveris. It is my pleasure to be hosting this joint webinar brought to you by Abveris and Katera. Today's speakers are Kobe Souders, the CSO of Abveris, and Noah Dito, Technical Product Manager for Katera. In just a minute, Noah and Kobe will speak on the topics of accelerated antibody discovery using high throughput and high content characterization tools, including the Katera LSA platform. Before we start, here's a couple very quick housekeeping items for the audience. The webinar is going to be split into 3 sections. The first section will be a presentation by Kobe on the recent Abveris discovery campaign targeting SARS coronavirus 2 spike protein. In the second part, Noah will present the in-depth technical features and applications of the LSA platform. There will be a live Q&A at the end to address questions from the audience. Please feel free to type your questions in the Q&A box now and during the Q&A section. We'll try to answer as many of them as possible given the scheduled time. Abveris and Katera will also follow up for questions that we don't have a chance to address today. We'll make the webinar available on our website within 48 hours, typically. If you want to rewatch it or want to forward that to your colleagues for the reference, you are sure to have access to it very shortly. A little bit of introduction to the speakers. Kobe is the CSO of Abveris. He provides scientific leadership and develops innovative strategies for antibody discovery. He got his PhD in cell and molecular biology from the Texas A&M University. Before Abveris, he was directing antibody discovery and engineering programs at Mass Biologics and Canuus Bio. Noah has been with Katera since 2014. Before Katera, Noah was with Bristol Myers Squibb for almost a decade, supporting drug discovery and early clinical development work. Noah earned his Master's of Science from the Penn State University, where he developed chromatography-based methods to isolate biomarkers in dengue infection. He also holds an MBA from West Chester University. So without further ado, I would want to pass the mic to Kobe to start his presentation. Kobe, whenever you're ready.
Thank you, Gary, and thanks to everyone for attending. And I'm happy to present the data that details our anti-COVID internal research campaign we completed last year at Abveris. We took a very function-forward and rapid antibody discovery approach for this project. Both are features that are key in the need for antibody discovery throughout the pandemic. It's also an antibody discovery model we feel works very well across a broad range of target classes, not just for rapid antibody discovery response to infectious disease targets. And I'll start by introducing the main factors that we feel are most important when designing an antibody discovery workflow. And these include antibody diversity, depth of screening, and speed. When we refer to antibody diversity, it's important not only to have large starting repertoire, but also to capture the full repertoire by eliciting a robust immune response, and then to recover a diverse number of B cells in the downstream screening process. But at the same time, the screening resolution has to incorporate enough complexity to identify the most functional clones early in this discovery workflow. And both of these factors need to be completed in a rapid timeline. And this depends not only on the actual time that it takes, but also how many clones can be screened in this defined time frame. In combination, this allows us to efficiently find the most relevant and rare functional hits. At Abveris, we combine advanced immunization techniques with our proprietary hyperimmune mouse platform to effectively generate a diverse starting repertoire, even to the most difficult target classes like high homology or self-targets, non-immunogenic epitopes, or complex cell surface proteins. Then, to mine the immune response, we've developed specialized hybridoma and single B-cell techniques paired with optimized high-content characterization. And this will be the main focus of this talk, to highlight how quickly and accurately we can triage a panel of hits to lead candidates. For the experimental design in this campaign, we leverage both a single B-cell cloning and accelerated hybridoma workflow. Both include rapid immunization strategies followed by primary cell harvest from tissues of interest. For the single B-cell workflow, screening is performed on the Berkeley Lights beacon in just 1 day, followed by sequencing and recombinant expression. For hybridoma-based discovery, colonies are screened by ELISA and Octet VLI within just weeks after fusion to select top candidates for parallel sequencing and culture scale-up. Top candidates from both single B-cell and hybridoma screening are then fully characterized by Katera SPR for affinity, blocking activity, and epitope mapping. Then combining the biophysical properties with in silico sequence analysis, we can select fully validated lead candidates within just a couple of months after initiating immunizations. We also use 2 different mouse strains to take advantage of the benefits of each one. The Alloy GK mice provide a wild-type-like immune response, but with the advantage of producing fully human antibody variable regions, while the Diversimab mice from Abveris produce murine antibody sequences, but elicit an enhanced immune response to broaden the epitopic diversity and ensure valuable regions of the antigen are effectively targeted. With our immunization strategies, we're specifically targeting the S1 domain that contains the ACE2 receptor binding domain, and both strains responded well, with detectable serum titers even at a greater than 50,000-fold dilution, giving us confidence that a robust anti-S protein B cell population was activated. In the single B cell workflow, plasma cells were loaded onto OptoSelect 14K chips using the Beacon, which uses optoelectric positioning to guide single cells into individual nanopens. Typically, we load more than 10,000 single cells per chip and can run multiple chips per day to have a total throughput of over 40,000 single B cells screened in a day. After cell loading, various capture and detection reagents are imported into the channels above the pen, and antibodies secreted from the plasma cells diffuse out of these pens and into the channel, forming blooms during the time-lapse imaging when they bind to the target reagents. And because the assay reagents are separate from the B cell, we can flush the reagents from the chip and import a second assay with all new reagents to repeat multiple sequential assays. Once B cells of interest are identified, we again use OEP to export the cell out of the well and into the channel, and then flush the channel into a well of a PCR plate for downstream sequencing of the heavy and light chain variable regions. For this particular study, we ran 2 sequential assays. In the first, we were looking to identify high affinity candidates in the low double-digit nanomolar or better range to the monomeric S1 domain of the spike protein. By capturing secreted IgG on anti-mouse polystyrene beads at the mouth of the pens, we detected total IgG with an anti-mouse secondary antibody in the FITC channel and positive binding to the S1 protein that's directly labeled with Lexaflor 647 in the Cy5 channel in a multiplexed assay format. Then, after flushing the channel, a second mixture of assay reagents was imported, this time with the S1 protein pre-complexed with a molar excess of ACE2 protein. So in this format, a lack of S1 binding signal indicated competition between the antibody and ACE2 for S1 binding, which identifies potential blocking candidates. Of course, it's also possible to label the ACE2 protein and identify antibodies capable of binding the S1 protein and displacing the ACE2, but that format wasn't run in this example that's shown here. On the hybridoma side, following a high throughput and efficient electrofusion process, thousands of colonies were screened by ELISA using various liquid handling platforms, and clones showing reactivity to the S protein, but not to an irrelevant counter screening reagent were pulled forward for secondary screening confirmation. Then, following the ELISA screening, we performed a single point kinetic analysis against the trimeric S protein to determine avid affinity using the octet. Recombinantly expressed antibodies from the single B cell workflow were already available for screening, so they followed the same characterization cascade. And out of the hundreds of binders that we discovered, the early triage criteria defined here identified dozens of top candidates to select for full validation, which included various SPR-based assays on the Cartera LSA. A representative subset of these clones is shown here, and full monomeric affinity to the S1 protein was determined in a 6-point concentration titration series. Overall, the Alloy and Abveris mice had very similar affinities, with clones derived from the single B cell workflow showing roughly a 2-fold affinity advantage over those from the hybridoma workflow. And this is also in line with previous head-to-head studies we performed where we select for average higher affinity on the Beacon. Next, a blocking assay was performed to identify clones with potential neutralizing activity. In this sequential format, an experimental antibody was pre-bound to the S1 protein and then exposed to the ACE2 protein. The amount of ACE2 binding was quantified and reported as a % increase in binding. And candidate antibodies capable of blocking the S1 ACE2 interaction showed negligible ACE2 binding like clone 11955 shown in yellow, but in contrast, clones like 12637S1 in green capture the S1 protein at a non-blocking epitope and allow the S1 ACE2 interaction to occur. Now, I'll pause here and take a minute to talk about the important comparison that we made with respect to the SPR formats as a part of this campaign. For all the Cartera studies that we performed, 2 different formats were used in head-to-head to validate the data from each one. In the most simplistic version, purified experimental antibodies were directly coupled to the chip surface prior to introducing analytes. And in this case, I'm showing the S1 protein binding to one immobilized experimental antibody before introducing a second candidate antibody in a classical epitope binning experiment. While this format is straightforward to set up, it does require purified antibodies in a defined buffer, which takes additional time and effort. Recently, a second assay format has been validated on the LSA, where antibodies in crude supernatant can be used. And this eliminates the time and cost associated with purification and buffer exchange. Now, this setup is slightly more complex because a capture antibody is immobilized on the chip surface to then bind the first experimental antibody, which then needs to be cross-linked, followed by an additional blocking step that's not actually shown in this diagram, prior to then introducing the analyte and second experimental antibody. In this study, we found the results from both assay formats were very similar though, and an extra 1 to 2 weeks could be saved on the overall project timeline by using the crude sample format. Analysis of the results from our epitope binning data in this format reveals a conserved epitope at the ACE2 blocking region, with some blocking candidates binding on the periphery of this core. Multiple other non-blocking candidates localized to different epitopes on the S1 protein, some adjacent and some distal to the blocking epitopes. And overlaying orthogonal data like avid and monovandal affinity suggests there's no clear correlation between affinity and epitope. Looking at another way to visualize the epitope binning data in the form of a community plot, we can clearly see 6 different distinct epitopes targeted by this subset of top candidates. And again, by overlaying orthogonal data onto the binning data, this time in the form of blocking clones in yellow and non-blocking clones in green, we can quickly establish which ones localize to the receptor binding domain to select high-value lead candidates. Now, to better illustrate how the crude versus purified samples compared in the SPR analysis, I'll highlight the binding profiles in more detail. So in this case, an example clone 16F2 has a nearly identical antigen binding profile when either purified antibody or supernatant is used. And when taking the rate constants into account for all clones, we see a nice correlation between the measured value for supernatant samples on the y-axis and purified material on the x-axis. Similar ranking and comparable values is evident for both the on and off rates and subsequently the final calculated affinity, which validates the use of either workflow to acquire accurate results when performed under these optimized conditions. For us, the option to use SUPs means we can characterize the data earlier in the workflow, and we have more flexibility in the sample requirements because we can be largely agnostic to attributes like buffer composition and antibody concentrations. Finally, combining the in-depth biophysical characterization with detailed sequence analysis allows us to select a few lead candidates from this larger pool. And when taking sequence similarity into account to define clonotypes, 3 unique groups of blocking clones emerged. And for a group with multiple clones, like the one with 4 similar candidates all binding to the core blocking epitope, we can incorporate in silico analysis of common sequence liabilities to further down select. And in this case, clones 21C3 or 11955 have very few predicted liabilities, making them excellent candidates for downstream development without the need for extensive engineering, which means that the time to clinic would be reduced for candidates that are like this. Interestingly, these 2 candidates do have the same sequence, but were each independently discovered with the hybridoma workflow for 21C3 and the single B cell workflow for 11955, which really validates both platforms for discovering high-value lead candidates. And to summarize the timeline for antibody discovery, the single B cell workflow can deliver sequences in as little as 1 month, with validated lead selection of recombinantly expressed antibodies taking just 1 or 2 months later. On the hybridoma side, the timelines are roughly doubled, with a total of about 2 to 3 months to sequence and 3 to 4 months from immunization start to recombinant expression and validation. but this can still be considered rapid when taking into account the large amount of data that's collected in just a few months. And now I'd like to thank everyone who contributed to the work at Abveris and Cartera, as well as Alloy Therapeutics, who provided the mice to help accelerate global COVID therapeutic solutions. And now I'll turn this presentation over to Noah, who will speak in more detail about the capabilities of the Cartera LSA platform on all of its different applications.
Thank you, Colby, and thank you for everyone who's online attending. My name is Noah Ditto. I'm Technical Product Manager at Cartera, and today I'll be talking about how the Cartera technology can be used to raise the bar for biologic discovery and characterization. So Cartera has really redefined the real-time label-free screening area in drug discovery and development. If we look at Cartera's technology, what it affords is 100 times more data in 10% of the time, using only 1% of the sample versus traditional platforms. So this is a significant leap in terms of what customers can do with a real-time label-free device in their discovery workflows. The Cartera's technology has been adopted across a wide range of companies. Listed here below is just an example. It really spans antibody developers, biotech, CROs, academics, as well as government research labs. The LSA is Cartera's flagship technology. What the LSA brings to the label-free real-time binding market is novel microfluidics that take SPR to a new level. So in the LSA, we have a multi-channel device, which is 96 channels that can dock on the sensor chip surface. And with that sensor chip fixed, the fluidics can switch over to single channel mode, where a single fluidic device docks on the surface and a single injection passes across all 384 locations simultaneously. So we can build arrays in the LSA of up to 384 proteins most commonly, but really any biomolecule can be attached to the surface. And then we're testing in single channel mode, one single injection across this array to really maximize the throughput on the system. So this really transforms what throughput looks like in terms of SPR. What controls the LSA is our Navigator software. The Navigator software platform is highly intuitive. Here's a screenshot just showing you kind of graphically how it lays out, very simple descriptions of where sample locations are going, pictorials of how the binding sensogram profiles are going to look like and the time that the user can specify for those. Even the most complex experiments, say a 384 by 384 binding experiment, which would last the better part of a week, can all be set up in about 5 minutes. And the arrangement of the samples and the plates is fairly agnostic in terms of the system's needs. So really the sample preparation steps are very simplified as well, compared to other platforms that sometimes have quite elaborate and specific needs for where samples are placed. So there's two main applications on the Cartera LSA that are most sought after when customers need to do real-time label-free screening in a high throughput manner. The first of those is epitope characterization. So really epitope characterization can span a number of different activities, being epitope binning, where we're competing antibodies to determine whether or not they share the same epitope on a given antigen, where we can compete antibody binding against a peptide array, and this is typically referred to as epitope or peptide mapping. So we're looking for binding sites to specific peptide residues and regions. And the last is mutant mapping, where we're taking an array of antibodies and presenting injections of mutant forms of the antigen across that as another way to sort of map more locally where the antibody epitope is located. Today, I'm going to focus on what is our core epitope characterization workflow that virtually every customer uses on the system because it is so high throughput compared to other platforms, and that is epitope binning. So just to step back and kind of look at the big picture, really what the LSA affords is faster and earlier epitope binning. This is just an example manuscript looking at over 100,000 interactions, 327 antibodies, and only using 5 micrograms of each antibody. So this is the type of workflow that can be now placed very early on in the discovery process, where typically This would not be allowed with other platforms. So really transformative in the space of epitope binning. Customers get a lot more detail about their clones. They're not trying to take low information, low resolution assays and use those to cull clones out. They can carry those clones further with a high confidence of knowing where they bind and their interactions with the other candidates in the overall pool. Here is an example of how antibody epitope binning can be conducted. So it all starts off with using that 96-channel printing device that I mentioned earlier to array up to 384 antibodies onto our biosensing chip surface. We then switch to our single channel mode and inject antibodies one at a time in a serial fashion to determine whether or not they can form a trimolecular complex in the presence of antigen. So in this case, this is a classical binning format I'm showing you here. We have a binding of an antigen, and then we have an injection of two different antibodies. One of the antibodies sandwiches, we have a clearly positive binding response, and the other fails to sandwich, which we have a no response, basically. And from this, we can gather whether or not the epitopes overlap or not for these respective antibodies. Inside the software, we have proprietary technology that allows us to cluster the antibodies together in these epitope networks. So we show antibodies having relatedness by having these sort of colored clustering or groupings. Lines between antibody nodes represent blocking relationships, and then the lack of lines between them represent, the ability to sandwich or no evidence of competition in this assay format. So this is a very sophisticated and sort of groundbreaking way to visualize epitope relationships among antibody candidates. And then what our software also affords is the ability to bring in additional data in the context of epitope into the application. So the application itself can have additional data pasted in, and then it's simple enough to display it in the context of all these networks. So it's a very intuitive workflow, very graphical, and really powerful in representing the antibody relationships and really what epitope means for the candidate overall. And just diving down a little bit more into those epitope networks, they're very fascinating in that you can start at a sort of granular bin level epitope network level, which is where we take the antibodies and we simply compete them, and in a very objective manner we just say, are you identical or not? In this case, we can see these shared groupings of antibodies. For example, this red group in the upper right here, these would all be identical in terms of their epitope because they're all competitive in the assay, so we would consider them identical for our purposes. And using hierarchical clustering, we can then say from these bin level epitope networks, we can take this to a community level epitope network and say that although there might be a few small differences between some clones, we're generally going to say that these antibodies are probably very similar in their epitopic footprint. So we can see on the far left here, this initial group in the upper center being similar, but not necessarily all clustering together, even though there is some competitive relationships. Once we do some clustering analysis, we can see that by and large, they are virtually identical, and we can therefore cluster them. This is a way to sort of move out of the detail and the noise of the assay to a level that sometimes helps with decision-making and driving distinct bins that can be used for downstream candidate selection. So I mentioned this a little bit previously. What does epitope offer besides just simply clustering antibodies? Well, there's this ability then to bring in orthogonal data into the assay. So this is really powerful. A lot of customers love using this because any attribute of the antibody that you have on hand, affinity, which you maybe derived already because you had done a kinetics experiment, blocking neutralization, what type of model species antigens are reacted against by the antibodies, library sources, expression levels, even sequence nuances can all be taken into the software and displayed on top of the epitope clusterings. So it's really, really powerful to understand collectively what attributes maybe drive selection for certain epitopes, or possibly even within a group of very similar behaving antibodies, find distinguishing factors that allow you to pick a single representative clone out of that cluster overall. Now, moving on to our kind of second major application on the LSA platform is kinetic characterization. So this would be assays looking primarily at binding kinetics. So we're looking at the on and off rates of the antibodies against antigens, In some cases where necessary, there may be a choice to do steady state affinities where we're not looking at the on rates themselves, but rather looking at it as a function of response versus concentration. But by and large, kinetics via measuring on and off rate is the most typical workflow done on the system. So truly high throughput kinetics. This is kind of a screenshot of 384 unique epitope measurement, or excuse me, kinetic measurements. You can see here that we're getting a wide range of affinities all measured in a single experiment. This entire experiment probably only takes about 6 hours. So we're measuring 384 affinities, one run, 7 micrograms of antigen consumed, and we get an 8-point titration. So really, really detailed characterization of clones. You can imagine that it's easy enough to multiply this and get up to close to 1,000 clones in a single experiment, because on the LSA we have 3 384 well plates. So there is the ability to take screening to the primary stage and really make sure that we get the most information per clone before starting to whittle down that list of candidates. So here's an example using single chain variable fragments raised against cars to show how kinetics works. So we have 384 clones arrayed on the sensor chip. These are most commonly captured so that we get preferential orientation of the FABs towards the solution phase and the subsequent antigen that comes in binding. Then we inject a titration series of antigen, or in this case, tumor receptor, across the array to see the binding on and off rates as a function of concentration. From there, we then take kinetic rate constants using a fitting model. So we have our K on, our K off, and then our calculated affinities. And what's really great about these assays is, in addition to simply measuring and getting kinetic rate constants and calculated affinities, we can actually include additional receptors, in this case of SCFEs for CAR purposes, and look for specificity as well. So the same array that you're using to measure affinity of your target antigen, you can also use by rearraying it and testing over and over again to check that you have the specificity you need, which is particularly important in the application of CARs. And in the kinetic software, really everything again is very much graphical and allows the user to explore the data, much like how Epitope has its network plots. This is just an example from the kinetic software of an isoaffinity plot that plots on rate versus off rate. And then we have on our diagonal here, we have the affinities that are calculated from this and sort of distinguishing bars. And as you hover over in the software, you can see individual sensograms for each of these. So again, a really easy way to visualize how your affinity distribution looks, and with the ability to label subsets of the data in the software, maybe particular clones from a certain source or any other attribute about those clones, you can actually sort of see affinity in the context of other pieces of data as well. So again, really powerful, easy to visualize, and all kind of baked into the software is ready to use rather than having to move on to different applications to perform these analyses. So big questions that comes up is, you can screen really fast on the LSA, that's great, and it can be done very early, and we're getting real-time binding data, but is it accurate? And the short answer to that is yes. There was an excellent paper published just last year by groups at Adimab and Amgen, kind of comparing the landscape of label-free biosensors. And among other things they determined in that exercise was that on the LSA, we get excellent agreement with gold standard platforms in terms of our measured rate constants and calculated affinities. And then the great part about that is then we're not trading throughput for accuracy, which is usually a typical trade-off in a lot of technologies. So really what that means in the big picture is that we're measuring affinities very early on in the discovery process. You can have a high level of confidence that those values determined then will hold throughout the transition to development, oftentimes where some of the early assays historically need to be repeated because we're changing platforms and there's discrepancies between low resolution screening and then the higher resolution screening towards the back end of discovery or as development kicks off. So again, lots of confidence in your measured affinities. So really, what does this mean for the researcher doing a study like this, where they're looking, both at affinity and epitope, it really drives decision making. This is a manuscript from Lilly, published this past year, looking at COVID therapeutics, and they effectively utilize the LSA to its full capabilities in this particular exercise, looking at affinity, understanding not only how tightly it binds, but disruption of ACE2 binding, which is particularly relevant for COVID therapeutics. And then also looking at epitope, looking at the breadth of the unique epitopes that are getting elicited during this natural immune response. So these were B-cell derived antibodies. Then also looking at whether or not the neutralizing antibodies had relationships to epitope, and really painting the overall picture of what these antibody candidates look like in terms of where they bind and how tightly they bind. So this is wrapping it all together. And really the icing on the cake for this particular example is that all of these studies were done in under a month in total, including the LSA data, which was collected, everything you see here, in about a 6-day span, which is incredible. I mean, when we're talking traditionally that these activities take months to years, we're condensing everything down into about 1 working week to collect a data set like this. So we have a huge breadth of information about the candidates, excellent rationale and data-driven approach to select the candidates, and all done in an extremely rapid and really record-setting manner. So the one last thing I'd like to touch on is kind of an exciting new area that the LSA technology is being utilized for, again, in this fight against COVID. And this is looking at the ability of immunoglobulin immune response to bind RBD in a 96 parallel manner from patient sera. So really exciting work here. There was a group, academic group out in the Netherlands that partnered with us and was able to generate a really cool data set showing that we could track the strength of binding. So that's the ability of the antibodies to bind and their rate of their off rate against the RBD binding. And then coming in after that, looking at whether or not there's certain isotype profiles that are unique among the infected individuals. So we're looking at IgM, IgG, and IgA binding, in addition to the strength and binding. So really powerful data set. Instead of just saying whether or not there's presence or absence of an antibody response, we're getting that plus the overall off-rate effect of the antibodies as time progresses and as you would expect the immune response to mature from an affinity perspective. All right, with that, I'd like to again thank all the attendees for taking the time out of their day to listen to our presentation. Really anything about Cartera's technology, LSA or otherwise, software, hardware, what have you, applications, feel free to reach out to us. You can use the chat to send questions during this webinar, or else contact me directly at nditto@carterabio.com, and I'd be happy to discuss further with you. Thanks again.
All right. Thank you, Noah. It's time for us to address questions from the audience now. And we have a lot of interesting questions coming in. The first one is for Kobe. You mentioned a lot of opportunities for improvement of the timeline. Would you leverage what you learned in this campaign to respond in another viral outbreak in the fastest timeline possible?
Yeah, that's a great question and a very important one too. So a few things that I mentioned within the webinar would be how to leverage the Cartera using crude samples. So that's something that you could do to save several weeks on a campaign, but still be able to capture the full value of that validation process to pick high value lead candidates, not just candidates, but it does save several weeks on that timeline. Another opportunity would be if you did want to proceed with mouse immunizations, you could optimize that timeline and again, use a rapid immunization approach to still elicit a diverse immune response and get high quality leads out of that. You'd also be able to use human B cells if the sample is available. So definitely several things, but the Beacon really does help accelerate that too by compressing all of the screening into a single day to select candidates.
All right. Thank you, Kobe. The second question is also for you. Do you believe that a mouse immunization strategy is better than a direct from human single B-cell screening approach for viral outbreaks? What are the pros and cons of both strategies, in your opinion?
Yeah, another good question. And again, kind of in response to the one that I answered for the previous one, both are absolutely valid approaches. So the direct from human approach absolutely gives you fully human antibodies up front. Often there's some difficulty in sourcing those samples though, so that would be pending availability of those samples. You're also limited to what response that particular individual has. So usually you don't have a large number of samples or the time to screen through a large number of different human samples that are available. And so you're hoping that the one individual or the handful of individuals that you're screening has the best response that you could get to find a lead candidate. When immunizing mice, you have that flexibility to elicit different immune responses based off of your immunization approach and your immunogen that you're using, and then different screening strategies as well. So you have a little bit more flexibility in that. Obviously, with the disadvantage of a slightly longer timeline by a few weeks, and also in using the humanized mice, you're also able to get a fully human antibody up front, which will accelerate that downstream therapeutic discovery timeline. But murine antibody discovery is still a valid approach, and again, can sometimes give you higher quality candidates and more diversity to find a better lead candidate, as long as you are able to afford the time to then do the humanization and engineering that would be required to turn that into a therapeutic downstream.
It makes sense to me. The next one is for Noah. How would you screen for gamma receptor binding using the LSA platform?
Yeah, good question. I kind of highlighted in our discussion, epitope characterization and antigen kinetics, which obviously work through the FAB end of the molecule. But you could consider then neonatal receptor, FcRn, or gamma receptor studies, kind of in the same vein as a kinetics approach, a captured kinetics approach. And in that way, we would capture the antibodies via the FAB region in some manner. There's a couple of different ways to approach it. And then flow the receptors in solution across that array. So you can get quite a high throughput of screening, up to 384 antibodies against a single receptor at a time. So you get really good parallelized measurements. But it operates very similarly to the kinetic approach that I described in the presentation.
Okay, that sounds good to me. Here's a follow-up question regarding Noah's comment. So the LSA can screen 384 samples, but is it practical to use the LSA if you don't have that, high throughput requirement in some cases?
Yeah, and definitely. There's a diverse range just of our customer base. Some absolutely max out the capacity of the instrument, and many don't approach 384 unique species at a time that they have to screen. One of the great things you can do, though, and this is kind of new to the SBR space, is you build in replicates in your assay or sort of scout optimal conditions as you run the assay. So particularly for kinetics, for example, the amount of ligand you put down on the surface, the antibody, it's critical you get that density right in order to get the confidence you need for the kinetic measurements you take. So in that example, you might have a subset of antibodies, but you could put them down, capture them, say, at 3 different densities and run the experiment, and then just use the signals that are most applicable to your kinetic measurement. Likewise, you have the ability to build in replicates. So you're getting statistical confidence in the measures you're making in a single experiment instead of having to run the experiment over and over again in order to build that statistical confidence. So yeah, the short answer is definitely there's a lot of power to be had, even if you don't have 384 or more clones that are unique to screen at a time.
Yeah, thanks. The next question is for Kobe. So in your presentation, you use a recombinant protein assay, but is it possible to run an infectivity assay using pseudovirus on the Beacon platform?
Yeah, absolutely. That one is something that has been developed. Really, the short answer is that it was availability of reagents. At the time that we were screening this, the reagents were not available to us, but that is something that you can develop. It does take a little bit more time because you'd like to run that and QC the assay and validate it. So if you're working as quickly as possible in a situation like this for a viral outbreak, then an assay like a protein blocking assay is faster to validate and get up and running on the Beacon. And so you'd be able to find high quality candidates, as we showed in this webinar, still using a recombinant protein blocking assay and then validating those downstream off Beacon and pseudovirus assay. But definitely if the reagents are available and the extra few weeks for time to validate the assay can be afforded, then a pseudovirus assay is possible on the Beacon, as well as a number of other complex screening assays. That's one of the big advantages, is that there's a lot of flexibility in the design and what you can run and the breadth of data that you can achieve. So definitely possible.
Yep. Thank you, Kobe. The next one is actually for you as well. So in this campaign, how large was the overall number of cells you screened and total number of hits identified? Did you show the full data set or just a subset of what you've done?
Yeah, so in comparison to other campaigns we've run, this one isn't quite as large. So we screened anywhere between 10 to 20,000 total B cells. This was before we got a second Beacon, and so our throughput has doubled now. And this is actually was done in a single run, again, to kind of recapitulate the idea of being able to move as fast as possible with a single run and see what you could get on a per run basis. So definitely could be augmented in future outbreak situations or in standard campaigns. And this is not the full data set that we did actually find hundreds of total candidates. We're just showing some of the best that perform downstream here in these assays. So the dozens that we show here is a subset of hundreds of total candidates.
All right. So before we jump to Noah, I would ask Kobe one this question from the audience. So you mentioned that you use multiple strains of mice to help with diversity. How did the downstream diversity play out across the different strains you use in your study?
Yeah, good question. Some of that is captured in the data, but in general, again, using the Diversimab mice, we are able to see binding across multiple different epitopes. We do see broader diversity in the sequence that is recovered, so clonotype usage is broad. We still see a good usage within the humanized mice as well, but again, kind of as the strain suggests, Diversimab provides a larger diversity. But again, going back to I think one of the earlier questions that was asked, and different immunization strains immunization schemes, and one of the advantages of using mice over direct from human B cells is the ability to use these different strains, elicit a different response, and get broader diversity overall by using multiple different strains that did allow us to find the most critical epitopes. Whereas with human B cells, you're kind of limited in that response. And so we were able to find very good candidates by leveraging these different mice and finding hits to multiple different epitopes, which, by the way, also can help if you're making oligoclonal cocktails, right? So for those to be really effective, you'd like to find antibodies that bind multiple different epitopes that all perhaps could be neutralizing epitopes as well. And so it's important to get this diversity, and it's much easier to capture larger diversity when you use multiple mouse strains.
Yep, sounds good. So we have a question regarding the LSA platform for Noah. Is there a reason to perform full kinetics versus single point when doing primary screening?
Oh yeah, so I think the question is really, historically, if somebody was trying to gain throughput on a label-free device, they often do sometimes what is just a single point screen. They just measure a single concentration and try to estimate primarily from the off rate a rank order that they can extrapolate for affinity. And that's really just had been done historically, again, by the limitations of throughput technologies. So if we think about the LSA and how fast it is, and how one of the examples I showed in my slide was having a full 8-point titration series for 384 ligands in only a 6-hour run, you really don't have to go through those exercises of trying to shortcut some of the assay processes. So really on the LSA, I would say that the most appropriate manner is to run a full kinetic titration, kind of that's what Colby's showcasing in their workflow. There's no point in trying to screen and get less data. It really doesn't save you anything in the end. And the one point I made in my presentation about the high accuracy of the data is that once you've got that number, it's good, it's solid. It's not like you need to go back and revisit it again and rerun that measurement. So you've got the number, you've got it screened early, and you've got as much candidate information as you can gather at that point to really make sure the decisions downstream are well informed.
All right, sounds good to me. The next question is for Noah as well. When doing serum serum studies like cited with the COVID serum work in your talk, are there any special requirements in assay design?
Yeah, so generally serum's not a problem. We do recommend 0.2 micron filtration just to make sure there's no particles or anything in the sample type. But we typically do a dilution, I believe in those particular studies in that manuscript was around a 1 to 100 fold dilution. So we're fairly dilute. About the only thing you may see is a little bit of non-specific binding sometimes with serum. That's usually just resolved by having a PSA as part of the running buffer to mitigate any non-specific binding of serum components to the surface, but otherwise, generally serum, given the level of dilution it has and just some best practices and monitoring for non-specific binding, is really acts as any other analyte would on the system.
All right, thank you, Noah. Since we're running out of time, so I will ask one last question, and we're going to wrap up today's webinar. This is for Noah. Is it possible to combine binning and kinetics measurement into one single run?
Yes, it is possible to do that. There's a little bit of balance of what you're trying to achieve. So typically for kinetics, most commonly customers would capture the antibodies. The example Colby gave was capturing directly at a crude material, enriching them, and doing your kinetics that way, as opposed to having to then do a separate purification step. That's usually pretty good, but depending on what you're doing with the binning side of the equation, you might need different ligand densities for the two of them. So for kinetics, we obviously want a very low ligand density, a very low amount of antibody on the surface to get the highest quality kinetics that we can get. Whereas in binning, we're running a lot of repetitive cycles where we're generating the surface and removing the antigen that's bound to test the next trimolecular complex formation in the next cycle. So we do have to increase the ligand capacity a bit. So it kind of, you have to sort of balance the two of those. Sometimes if customers may want to set them up separately and run them, but you can combine them if you're willing to maybe make those trade-offs, saying I'm willing to get maybe a little bit of non-ideal ligand density for kinetics in order to make sure that I can combine the binning. Maybe for your timing purposes, that's a necessary or reasonable trade-off. So that's probably the only thing to consider is the ligand density components of each assay.
All right, so thank you, Noah. Thank you, Colby, for answering the questions. And time's up, so we have to say goodbye. But please bear in mind that we're gonna reach out to questions that we don't have time to answer today. And as a reminder as well, we're gonna make this webinar available on the website shortly, in 40 hours usually. So please visit our website to get a link to it if you want to rewatch it or share it with your colleagues afterward. And that's it. Thanks for joining us today, and bye-bye. Look forward to seeing you in another webinar.